In this video, we will discuss about the types of electrodes. Types of electrodes. There are three types of electrodes. They are first one, microelectrodes. Microelectrodes are used to measure the bioelectric potential near or within a single cell. These are also called as intracellular electrodes. Second one is depth and needle electrodes. These are used to measure the bioelectric potentials of the highly localized extracellular regions in brain or bioelectric potential from a specific group of muscles. The third one is surface electrodes. These are used to measure the potentials available from the surface of the skin and are used to sense the potentials from heart, brain, and nerves. Microelectrodes. The microelectrodes are divided into metallic and non-metallic. The microelectrodes should have a smaller diameter, and during insertion of the electrode into the cell, there will not be any damage to the cells. When a microelectrode is used to measure the potential of the cell, it is located within the cell, while the reference electrode is situated outside the cell. The size of the electrode is determined by the size of the cell. Since the size of the cells is about 50 microns, the diameter of the tip of the microelectrodes is ranging from zero point 0.5 to 5 microns. Metal microelectrode. Metal microelectrodes are formed by electrolytically etching the tip of a fine tungsten or stainless steel wire to a fine point. This technique is known as electropointing. The metal microelectrodes are coated almost to the micro tip with an insulating material. The figure shows the position of the electrodes and its equivalent circuit. Since the measurement of the bioelectric potential requires two electrodes, the voltage measured is the difference between the instantaneous potentials of the microelectrode and the reference electrode, and it is a sum of three potentials such that EA metal electrode electrolyte potential at the microelectrode tip, EB reference electrode electrolyte potential, EC variable cell membrane potential. The equivalent circuit RA denotes the resistance of the connecting wire, which is negligible. RS denotes the resistance of the shaft of the microelectrode, which is also negligible. RFA, RWA, and CWA constitutes the impedance of the microelectrode tip intracellular fluid interface. RIN is the resistance of the intracellular fluid. Similarly, RB is the resistance of the wire connected to the reference electrode, which is negligible. RFB, RWB, and CWB constitute the impedance of the reference electrode extracellular fluid interface, and REX is the resistance of the extracellular fluid. CD is the distributed capacitance between the insulated shaft of the microelectrode and the extracellular fluid. The capacitance between the tip of the microelectrode and the intracellular fluid is negligible because the potential difference across it does not change. The equivalent electrical circuit can be approximated as shown in the diagram. Since the area of the reference electrode is many times greater than the metal electrode's tip, whose area of cross section is very small, its impedance is negligible. Because the impedance of the microelectrode tip is inversely proportional to the area of the tip and frequency. When the electrode output is coupled with an amplifier, the low frequency components of the bioelectric potential will be attenuated if the input impedance of the amplifier is not high. Thus, when the input impedance of the amplifier is not high enough, it behaves as a high pass filter. Micropipette. The non-metallic micropipette consists of a glass micropipette whose tip's diameter is about 1 micrometer. The micropipette is filled with an electrolyte, usually 3 molar KCl, which is compatible with the cellular fluids. A thin flexible metal wire from chlorided silvers, stainless steel or tungsten is inserted into the stem of the micropipette. The friction between the wire and the stem of the micropipette and the fluid surface tension hold the micropipette on the wire. The other end of the metal wire is mounted to a rigid support, and the other free end of it is resting on the cell. The equivalent electrical circuit can be drawn as shown in the diagram. From the figure, EA, the potential between the metal wire and electrolyte filled in the micropipette. EB is the potential between the reference electrode and the extracellular fluid. EC is the variable cell membrane potential. ED, potential existing at the tip due to different electrolyte present in the pipette and the cell. Then E is equal to EA plus EB plus ED. RA denotes the resistance of the connecting wire. RFA, RWA and CWA constitute the impedance of the electrode electrolyte interface in the stem of the micropipette. RT is the resistance of the electrolyte filling the tip of the micropipette. That is the resistance of the tip, which is very large. Rn and Rex are the resistance of the electrolyte inside the cell and the electrolyte outside the cell, respectively. RFB, RWB, and CWB constitute reference electrode electrolyte interface impedance. RB is the resistance of the wire connected with the reference electrode. CD is the distributed capacitance existing between the fluid in the pipette and the extracellular fluid. This equivalent circuit can be further approximated as shown in the second figure, where CD dash is the equivalent of distributed capacitances when the micropipette is coupled with the amplifier terminals A and B, then the membrane potential EC is coupled with it via a high series resistance RT and a moderate shunt capacitance CD along with electrode potentials. The impedance of the electrode places a limit on the response time of the circuit such that it behaves as a low pass filter when the input impedance of the amplifier is not high enough.